Welcome back to my channel Knowledge Chronicle.
Today we're diving deep into how biotechnology is transforming the world of medicine, from manufacturing life-saving insulin using bacteria to curing genetic diseases through gene therapy and diagnosing deadly infections even before symptoms appear. So if you're curious about how tiny molecules and big ideas are saving lives, stick with us till the end. Let's start with a simple question. How are today's medicines better than before? The answer lies in recombinant DNA technology. It allows scientists to manipulate genes, insert, delete, or modify them to produce specific proteins or hormones inside microorganisms like bacteria or yeast. Thanks to this, we now have mass production of safe, effective therapeutic drugs, many of which are identical to human proteins. Earlier, proteins extracted from animals often caused allergic reactions because they were slightly different from human versions. But recombinant proteins, made using human genes inserted into microbes, are much safer. Globally, around 30 recombinant therapeutic drugs are approved for human use. In India alone, 12 of them are available and widely used. One of the most groundbreaking examples is genetically engineered human insulin. Let me explain how revolutionary this really was. Before biotechnology, insulin for diabetic patients was extracted from the pancreas of slaughtered pigs and cattle. Although similar, this insulin sometimes triggered immune responses and allergies in humans. Now imagine this. What if a bacterium like E. coli could make human insulin? Well, that's exactly what scientists did. In 1983, an American company named Eli Lilly synthesized two separate DNA sequences corresponding to chain A and chain B of human insulin. These are two small protein chains that form insulin, connected by disulfide bonds. Here's the catch. Insulin in our bodies is first made as a prohormone called proinsulin, which has an extra piece called the C-peptide. This C-peptide is removed to make mature, functional insulin. Eli Lilly introduced the two chains into plasmids of E. coli bacteria. The bacteria made the A and B chains separately. These were later extracted and chemically combined by forming disulfide bridges, thus creating fully functional human insulin. And by the way, why can't insulin be taken as a tablet? Because it's a protein, and our stomach enzymes would digest it. So, insulin must be injected directly into the bloodstream. Let's talk about something even more futuristic. Gene therapy correcting diseases at their genetic roots. Imagine a child born with a defective gene. Instead of just treating symptoms, what if we could fix the gene itself? That's what gene therapy aims to do, by inserting a normal, functional gene into cells to replace or compensate for the faulty one. The first clinical gene therapy happened in 1990. A 4-year-old girl had a rare genetic disorder called ADA deficiency, where her body lacked the enzyme adenosine deaminase, which is crucial for a working immune system. Here's what doctors did. First, they took lymphocyte cells from her blood. Then they introduced the functional ADA gene using a retrovirus as a vector. Finally, they reintroduced the modified cells back into her body. However, since lymphocytes don't live forever, the treatment had to be repeated regularly. A better and more permanent method would be to introduce the gene into embryonic cells or bone marrow stem cells so the corrected gene stays for life. Now let's move to something critical. Early diagnosis of diseases. Why is it important? Because many diseases like HIV or cancer become dangerous long before symptoms appear. Traditional tests using blood or urine often detect the disease too late. But modern techniques like PCR and ELISA allow us to detect diseases, even when the pathogen is present in tiny amounts. PCR, or polymerase chain reaction, works by amplifying the DNA or RNA of a virus or bacteria. For example, even if there's just one molecule of HIV DNA, PCR can make millions of copies, making detection easy. It's now routinely used for AIDS testing and cancer mutation detection. Another method is ELISA enzyme-linked immunosorbent assay. This technique is based on antigen-antibody interactions. Let me explain with an example. If someone has a viral infection, their body will produce antibodies against it. ELISA can detect either the antigens from the virus or the antibodies made by the body, helping diagnose infections like HIV, hepatitis B, and even COVID-19. Another precise method is using radioactively labeled probes. A single-stranded DNA or RNA tagged with a radioactive marker is allowed to bind to its complementary DNA in a patient's cells. If it doesn't bind, that means a mutation is present, and that helps in diagnosing genetic disorders. So to sum up, biotechnology has completely changed the face of modern medicine. Recombinant DNA technology enables mass production of safe, human-like medicines. Genetically engineered insulin gives life to millions of diabetics. Gene therapy offers hope to children born with genetic diseases. And powerful techniques like PCR and ELISA help us detect diseases faster than ever before. These are not just technologies. They're life-saving revolutions, crafted by science for humanity. If you found this video informative and interesting, then your one like, share, and subscribe will help us create Chronicle, where science meets curiosity. See you in the next episode.